This is the Convergent Science Network podcast. Leading researchers in the domain of neuroscience, brain theory, and technology are interviewed by Paul Verschure and Tony Prescott.
Yeah, all right. This is Paul Verschure with the Convergent Science Network. This is our BCPT School 2015. And I'm here with Brian Kolb, who this morning was in his lecture dealing with the whole question of epigenetics and how in some experience can affect our behavior even for a number of generations after us. So what is specific about this epigenetic perspective on behavior and changes in behavior?
Well, I think that historically people had this view of nature and nurture, and that most of what we developed, we learned, and it was sort of independent of anything else. And I think what we now know is that there's an interaction between the activity of genes in the brain and experiences. So specific experiences turn different genes on and off. And when they're doing that, it alters how the brain responds to other experiences. So, for example, if you're taking a drug, and let's just say it's nicotine, the nicotine is changing the expression of genes in the brain, and the job of genes is to make proteins. So what the proteins they're making are different. And so the organization of synapses, circuits, is altered by the drug in an indirect way. But it turns out that it depends on which genes are already on and off. So the baseline gene profile is going to actually influence how experiences do this.
Mm-hmm. But now, in some sense, anything that a cell does, or almost anything, will have an impact on gene transcription.
Yes.
So, however, there's a certain subset of gene transcription that are of specific relevance to this question of the epigenetics, where as I say, the gene expression and environment are coming together. So is there a specialized, if you want, system that is supporting these epigenetic changes to behavior?
That's the general belief. But if you now say, where is that system and what is it, we don't know.
Okay.
Yeah.
But that's sort of the assumption that we
and it may be that it's microRNAs that are changing and non-coding parts of the gene that are changing and controlling all of this, but we're still pretty naive on how that works.
Mm-hmm. Right. So, can you give me what's a typical example that you would have in mind that really expresses most clearly these epigenetic impacts that we have on the phenotype?
Well, if you look, we've looked a lot at stress and various kinds of stressors because stress produces such big changes, and these changes persist. So we know that changes actually can cross at least 4 generations. If you experience stress in utero, so gestational stress, we know that there are changes in a whole bunch of things, and these changes persist. So we're looking at different kinds of stressors and trying to compare them and see if we can get some understanding as to which pathways are actually changed by the stressors. So basically we're taking a sledgehammer and trying to see, okay, if you use a sledgehammer,
mm-hmm.
what do you get? Rather than something more subtle like social interaction, which would be much harder. I'm sure it's doing things. And our example of play behavior is one example where the way in which animals play is going to alter gene expression and it's going to alter synaptic organization, but it's much more subtle.
Mm-hmm.
But
stress. Now, if we talk about these kinds of changes, how would you measure those? If you now say, okay, there's some environmental, let's say a stressor that is impacting a mother rat, now the offspring will be changed as a phenotype. What are you looking for in that phenotype?
So there are various things you can do. So the first measure, so when the rubber hits the road, this is all about behavior. And so we're looking for changes in behavior. If we find none, it might be that our measurements are too simple, they're not sophisticated enough, or it may be that there aren't any changes. So at that, first we're looking for changes in behavior. When we see them, we know we're on the right track. Then the second thing we're interested in is, okay, can we see changes in the circuits in the brain? And where are those changes in the circuits? Because you can't look at gene expression everywhere. It's just not practical. So you're looking for a needle in a haystack. So we look at using brain staining, using a Golgi technique. We can look at the changes in the number of synapses on cells. Once we see a region that looks like it's showing significant changes, we then can zero in there and say, okay, there's where we'll take our tissue and do our gene chip arrays or whatever it is we're doing to try and identify the genes. Now, the problem is the ideal thing would be to find one gene. Well, that's not going to happen. You're going to find cascades of genes. So we often are finding 1,000 or 1,500 genes. That's useless. So what you have to do is to see, okay, these are going to be related to one another in some way. And so we're looking at trying to get some sense of what the pathways are. Are they all related to the production of proteins related to synaptogenesis or neurogenesis or some other thing? So that's the process. Behavior, and then where in the brain are things changing, and then drill down with your genes. And we start with looking at global methylation. So what methylation is telling you is that if you have a change in methylation up or down, you have more or less genes being expressed. The more methylation, the less gene expression. Once you've seen that, then you can say, okay, that's just a clue that something's going on, but it doesn't tell you much. It just tells you something happened. Then we can look at the actual genes themselves, because it's expensive and you don't want to waste time looking at in places where nothing happened.
Mm-hmm. Right. So now, a first example that you emphasized a lot was the sort of the role of tactile stimulation as an epigenetic factor. So how does that play out exactly?
So just to give the background, if we take young animals and we use a little brush and we tactily stimulate them for 15 minutes, 3 times a day for 10 days, let's say, we can, and then wait till they're adults, we can see changes in their behavior. So we see enhanced cognitive skills, better memory, better learning. I don't know about perceptual skills. We haven't done that. But then we can look in the brain and say, okay, are there changes in synaptic organization? Yes. So where are they? And it turns out with tactile stimulation, that's pretty widespread. And then we can say, okay, what are the changes in gene expression? And can that give us some clues to what the tactile stimulation is actually doing? The advantage of that is if we now want to use that tactile stimulation as a therapy, let's say for an animal that was stressed, can we reverse the effects of the stress and reverse the effects of the gene expression? Well, if it turns out that it's totally different systems of genes, it's going to be a tougher sell than if there's some relationship between the two.
Mm-hmm. But now you also emphasized the release of FGF2 as a result of tactile stimulation. So why do you emphasize that factor so much?
So we started looking at FGF2 for other reasons, because we knew that in vitro it stimulates neurogenesis. And so we originally thought, well, maybe we'll be able to use FGF2 to stimulate neurogenesis, or maybe the tactile stimulation will increase neurogenesis. So that's why we started with it. And it turns out it did seem to be released with enhanced tactile stimulation, enhanced the release of FGF2. And so then we thought, okay, can we see a pathway in the brain related to this? Is there an increase in the genes related to FGF2 receptors or the production of FGF or whatever? So really, it's one of these things that's your favorite molecule by accident. But once you found something, let's stick with it, and we'll see what we can get from it.
Right. Okay. So because also other substances are released in response to tactile stimulation, right? Like endorphins, for instance.
Sure. And so are probably cholinergic systems in the skin that are related to pain and whatnot, right? All of those things are released too. And so we have looked at acetylcholine levels, and they do change too. But we haven't pursued that and haven't seen any evidence in the epigenetics that acetylcholine is really going to be a likely candidate for the big one, if you like.
Right. Okay. So now an important from there, so after tactile stimulation, but also identifying a possible pathway of then such an epigenetic channel, because that means the tactile stimulation is also then already, let's say, with the mother, right, the pregnant mother, and then the stimulation would then have an effect on the pups. So what's the behavioral impact that would have?
So, I mean, there's two issues here. One is, how do we know that the tactile stimulation hasn't changed the mother's brain and it therefore changes her behavior towards the pups, and that's what's doing it? We don't.
Mm-hmm.
But we do know that if we look in the brains of the pups, we see change in FGF2 receptors, for example. And if we look at their behaviors, we're going to see enhanced cognitive behaviors, enhanced motor behaviors, and not so much species-typical behaviors, but like play and so on. So we know that whatever the FGF2 is doing to the mum and the fetus is producing these behavioral outcomes.
Mm-hmm. Okay. So, but then actually you made quite a big jump, right? Because then we looked at the growth of vocabulary.
Yep.
In humans. So how is that? What's the effect that you observe there?
So the idea there is that experience early in life is going to make a huge difference to the development of language. I'll give you a simple example, and that is when you're a month old or a newborn, you can discriminate all speech sounds in all languages. And a few months later, you're losing that. So if you're only exposed to Dutch, for example, mm-hmm. you're not going to find Korean so easy. You're going to have difficulty discriminating those speech sounds, and the longer we go, the less able you are. So we're going to have that going on. The other thing we're going on is that exposure to words makes a huge difference to the development of cognitive abilities. But it's the exposure to words isn't just hearing the words, it's actually using the words. So some people will call this serve and return. So if I say, Paul, what did you do today? I'm expecting a response. Mm-hmm. If, on the other hand, you're listening to radio or television, you're not responding. And so you're not using language in the same way. So there's a really interesting experiment which children, and I forget the age, it was like 1.5 or 2, were either watching somebody teach them Japanese, or a Japanese person was in a screen and there was serve and return. So they had to say the sounds. And it turns out if they were actually interacting with an individual, there was a social aspect. The kids learned Japanese words, whereas the ones who were doing it on a television monitor didn't. Mm-hmm. So that serve and return seems to be important. So what is it about that? Well, there's a social aspect. And if you're actually teaching an infant things, there's normally a lot of contact. So you're getting tactile stimulation of them. They're sitting on your lap or your arms around them or whatever, mm-hmm. right? So all of these factors are going together. So then if you say, well, why is it that children who live in higher socioeconomic status houses have a larger vocabulary? Is it because their parents are smarter? No, their parents have a larger vocabulary, so they're exposed by age 3 to about 1 million more words than the kids in the lower SES families. Not 1 million different words, but 1 million more words total that they're exposed to. A lot of them are different words. And largely it's because for one reason or another, the higher SES families, there's more serve and return. Mm-hmm. So there's more discussion about things than there is in the low SES families, perhaps because the caregivers in the more wealthy families have more time, mm-hmm. or that there is a caregiver that's hired that's there all the time, a nanny or whatever it might be. Whereas in the less well-off families, they don't have that. Mm-hmm. Or they may just be interacting with siblings. And of course, siblings aren't going to have as large a vocabulary, and they may not serve and return in the same way as adults. Mm-hmm. And the effect of this is to get kids on a trajectory so that they're learning more and more words. And as I mentioned this morning, the kids who at age 36 months have a vocabulary that's about 3 times as big as the kids who are less well off, that difference just continues to get larger and larger and larger. And so by age 11, those kids in the high SES families have a larger vocabulary than the mums of the other kids.
Mm-hmm.
I also mentioned the fact that you could say, well, school is going to reverse this. So once they get to school, they're all going to be exposed to the same. That's true. But the less fortunate kids are way behind. And a study in New Zealand showed that after 8 years of school, it didn't make any difference. The kids who were low are still low. The kids who are high are even better. And so we need to do something in school to try and reverse this. And part of it may be to increase the amount of serve and return for these lower SES kids, more interaction to try and get them using language more aggressively.
But now you could also argue that you mentioned stress earlier, that these sort of lower socioeconomic status families have also higher stress. And
Absolutely.
That the real explanation is more at the end of stress than at the end of, let's say, exposure to language.
They're obviously intertwined. So stress is clearly going to be an issue. So one of the ways you could study it is retrospectively, in a sense, and that is look at stress levels in the two groups and try and tease that out. People are trying to do that. That and as well as diet and a variety of other things. Mm-hmm. One of the best studies on this is done in Cuba, and it's a social experiment. So Castro, it turns out, was really interested in children. So after the revolution, he changed the educational system, and they started pouring resources into children, and children had to go to, the mums, first of all, to what were called polyclinics. So they had to report monthly to this nurse, and if they didn't show up, they went and found them and brought them in. And the polyclinics are associated with schools. And so when UNESCO did their first studies on Latin American skills in kids, they compared kids in Cuba to kids in Chile, Argentina, Mexico, and so on. And what they found was that the kids in Cuba were significantly better in both literacy and arithmetic skills. So the question is, well, is it just that Latin America isn't all that good, and this little bit of extra experience in Cuba worked? There's two ways to look at that. One is to say, well, I'm from Canada, to say, well, how do Canadian kids do? We're probably better than the Cuban kids, and the answer is no, we're not. We're actually less good than the Cuban kids. Mm-hmm. Well, can you take what they did in Cuba and apply that in less advantaged places? So there was a study in South Carolina working with children from disadvantaged Black families. They used the same system, and they ended up with the Cuban scores. Similar study done in Mexico, same outcome. So it looks like pouring resources in early makes a big difference. And there's another UNESCO study, it was an OECD study, looking at literacy skills. And they identify 5 levels of literacy. Mm-hmm. And you're going to have literacy level 5 because of your education and experiences. But the average person doesn't. And in Canada, about 42% of the population is considered illiterate, even though they all went to school. So they're levels 1 or 2. In the United States, it's even higher, and in Britain, it's higher as well. The United States is about 56% is considered illiterate. So this is pretty scary. Mm-hmm. Well, let's look at other countries. So if you look at Sweden, it's about 30%. So are Swedes innately smarter than Brits or Canadians or Americans? I prefer to think not. What's the difference? They pour more resources, just like Cuba does, into early childhood development. Mm-hmm. And that seems to have a huge impact on language skills and subsequent literacy, which, mm-hmm. I mean, literacy has a huge impact on your health and your income.
Mm-hmm. But the consequence of that is also that maybe the real impact is actually before children go to school.
Oh, it is. Uh-huh. There's no question that it is. It's in those first 3 years, probably.
Right. So then what's the benefit of going to school?
Well, you're learning information, obviously. Mm-hmm. And you and I went to school for a long time, and we learned a lot of information. So why didn't the people who were in levels 1 and 2 benefit so much? They were behind the 8 ball to start with. And it wasn't that they were stupid, to use a sort of blunt term. They really were set on a trajectory that was disadvantageous. Mm-hmm. So the challenge here, and the Swedes have got it right, is to intervene really early. Mm-hmm. And in those first 3 years, and put the money there rather than trying to reverse things when the kids are 10 or 11 when it's too late.
Right. But then A key factor that you emphasize a lot is notion of stress, and also you looked a lot at stress in rats and how this then impacts their offspring, or how it impacts their mates.
Mm-hmm.
So you take a rat. I understand the main procedure to induce stress is to put them on a raised platform, and that's then sort of a predator anxiety that they are exposed to. That's a source of stress. And now you observed a number of interesting effects, right? So rats just on their own, rats that are exposed to that stress, what does it do to their brain and their behavior? Just if I take an adult rat and I would put it on this platform for a number of times.
Okay, so we've done that, just taking adult rats, and we increase their anxiety. We affect their ability to learn complex motor skills. We see big changes in the organization of synapses in prefrontal cortex and different effects on different parts of prefrontal cortex. And we see changes in gene expression. Now, it's a bit confounded because how do we know that the behaviors that they're exhibiting now, the anxiety, isn't producing the changes in gene expression? Well, they don't know that. Mm-hmm. There's no way for me to separate those two things, except by correlation and say the ones who are the most anxious, do they show bigger changes? Yeah, but that's still confounded. Mm-hmm. Yeah, so it's very difficult. But we do know that we can produce similar effects in adults as we can produce indirectly in the developing brain.
Mm-hmm. So now how do you assess anxiety in those rats?
So we can do it in a variety of ways. The simplest way is to put animals in a situation where they can hide and be safe, or they can go out and explore. And the inference here, or the implication, is that animals who hide are afraid, and the ones who go out and explore are not. Well, I think you mentioned in a question this morning that, well, how do I know that it just doesn't make them more active? So there's another test you can use. We haven't actually done this yet, but we're going to, and that is, it's called a burying test. So let's imagine you're in an environment and there's this little probe that comes out, and you go and you touch it, and you get a shock, and you're a rat. Not a bad shock, but like a carpet shock. Okay, so you get this shock. Animals that are anxious will bury that probe. They'll cover it up, and the more anxious they are, the more they cover it up. Animals that think it's trivial, for whatever reason, because they're on Valium or something at the time, go, never mind. Mm-hmm. Just avoid it. Right. So that's another measure of anxiety that we're gonna cover that up. Mm-hmm. And another way you could do this is to take animals that appear to be anxious, for example, in that plus maze, and give them anxiolytics and see whether or not that reduces the effect you've seen it does.
Right. But now, okay, so we have the adult rat, but now what you also told us, if I take this adult rat and I bring it back to its mate, they will actually communicate about their experience.
Yeah. So rats have a very complex set of songs that, I'll use the term loosely, songs that they sing. Pardon me. And the rat that's stressed will come back and sing a series of different distress songs. So life is crap songs. And the mate will sing back songs that are usually associated with happy things. Life is good. And this goes on for hours. And apparently, this is an inference, not proven, hearing this distress song for so long is stressful. And that affects the offspring of what we call the bystander animal.
Mm-hmm. Okay, but then what's the impact on the offspring? How strong is that impact as compared to the mother having been exposed to that stress herself directly?
I think the best answer is the effect is different. Mm-hmm. We see an effect. We see increased anxiety. We see reduction in brain weight. We see impaired motor skills. But in each case, it's not as big as the direct stress. It's a smaller effect. Mm-hmm. So the next question is, well, can you vary the intensity of stress given directly to the mom? And would you see, so for example, instead of putting them on that platform for 20 minutes, put them on the platform for 10 minutes. Would you see a different effect? And the answer is yes. Mm-hmm. So the intensity of the stress seems to make a difference. And so we're mimicking a lower intensity stress using the bystander stress.
Right, exactly.
Yeah.
But then the offspring will show also higher anxiety, as we saw earlier in the adult rat exposed to the stress?
Yes.
Or would it also show other kinds of behavioral changes?
We can actually look at behavior at about 9 days. And there's a variety of behavioral tests you can use to try and look at nervous system development. And it looks like rats exposed to that kind of stress are delayed in development by about a day. They're slower to develop some of the early behaviors. So we can see it right away. If we look in adults, we see changes in cognitive skills, motor skills in particular.
Mm-hmm.
Now, one of the questions we've been asking is, well, if you've had that early stress, how do you respond to stressors later? Is there any kind of inoculation effect, an advantage? And the answer is, it kind of looks like there is. That if you've had that early stress, that a later stressor, say as a juvenile, isn't as effective in changing your brain. It's like you've been inoculated against stress. So we're pursuing that now and varying the age of the second stress and the first stress and trying to see, okay, how does that work?
But that means you might have paid a price for that in terms of your cognitive abilities.
You may have paid a price for that, but you may have gained an advantage in coping skills.
Mm-hmm. Right, exactly.
Yeah.
But it's a trade-off. It's not something that comes for free.
Exactly.
And then that might mean that also the offspring, now the third generation, might also again be still under the influence of that stress that then their grandparent was exposed to. So how far down the lineage would this go?
Well, my colleague, Linda Metz, has been studying this, and we've done a couple of experiments with her, and we've gone to the fourth generation, and we can still see effects.
Mm-hmm.
One of the interesting effects is on the gestational age at birth of, say, the grandchildren and great-grandchildren offspring, and it turns out that those early stressors are actually changing the length of gestation by half a day or so. So to what extent is that the cause of all of this rather than the stress itself? Don't know. So these, you can see it starts to get complicated. But the reason that she's doing, I'm not interested so much in the gestational period, but the reason she's doing it is because in humans, it looks as though stressful experiences will do the same thing. They'll change. You get more premature babies. And so what's that doing?
Right.
Yeah.
But then do you see this wash out? So after how many generations would it be washed out?
Well, it has to wash out. I mean, logically, it has to wash out, and you're going to have regression to the mean. So Michael Meany at McGill has looked at it in a slightly different way. What he's done is say, okay, we don't stress anybody. We'll just look at the endogenous behavior of mums. And what he identifies is mums who do a lot of licking and grooming of their offspring and mums who don't do as much. So you have a normal curve. Let's look at the two tails. Can you breed for that? And it turns out you can't, that there's a slow regression to the mean. And why would you even care about licking and grooming? Because it's related to behavior, just like the tactile stimulation is related to behavior. And so, but over time, it seems to go away. And because we thought, well, we could just breed for this, and no, it doesn't work.
But now there must be a range of stress to which the animal is sort of genetically, let's say, prepared.
Yes.
That it can tolerate and it will not have these kinds of epigenetic knock-on effects.
Yes.
So how broad is that range?
Well, that's a good question. I mean, clearly, if you think about the old psychological inverted U function for stress, you need some stress or you're not awake, right? If you have too much stress, you're dead. And so where exactly in that inverted U function is the effect optimal? And when is it worse? Right. And so that's the kind of experiment that one needs to do to try and figure that out. We don't know that.
Okay.
But I'm sure that there's a level of stress that nature is expecting to encounter. There has to be. And so it's had millions of years to adapt to that and to expect it. So when we talk about brain plasticity, we talk about experience expectant. The brain is expecting certain kinds of experiences. And if it doesn't get it or it gets too much of it, it goes, whoa, what's going on? And you
But now the interpretation in this case is rather non-specific because we say, well, there's some environmental manipulation leading to stress on the animal, and the stress then has an epigenetic impact.
Yep.
But maybe with this manipulation, you could argue, well, maybe the impact is very specific. It is basically telling this mother rat directly or indirectly that this is an environment filled with predators.
Yes.
And that then the epigenetic effect is actually a very specific adaptation, which is weird. Let's say, stay hidden more, don't go out a lot, avoid open spaces. So what we then interpret as a non-specific impact of stress is maybe very specific adaptation to a world that is filled with predators.
Absolutely. And if you think about the Barker hypothesis, which is a nickname for that kind of idea, if you look at the onset of adolescence in animals who were stressed, it's earlier. So we see this. So why would that be? Well, they're going to have, if it's a dangerous world, you want to have your baby sooner. You don't have the luxury of waiting.
Mm-hmm.
And whereas the animals that are raised by moms who are very attentive, it's later.
Mm.
So again, it looks like it's adapting to the environment that's going to be there. Which adaptation is correct? Well, it depends on what the environment turns out to be.
Right, exactly.
Yeah.
So, but then there might also be the other extreme. If you have animals that have zero stress, like it's the Wall-E world. I don't know if you remember that animation movie of the humans all living in outer space.
Yeah.
Being fed high sugar or high glucose drinks all day long. Would you also see epigenetic change when you have a world that is really zero stress or like under stressed?
I would think you would, because using the experience expectant model, the brain is expecting stress.
Mm-hmm.
And in the absence of it, it's going to change. There's going to be some sort of change. Now, to do this experimentally would be a bit of a challenge.
Right.
But in principle, as a mind experiment, a thought experiment, that should be the case.
Right. So we have a bit of an insight now in the stress case, and then also these effects that it has epigenetically. But another manipulation that you have looked at is drugs, different kinds of drugs of abuse. Do you think the impact of drugs of abuse is let's say, similar to the impact that stress has, or is it?
So when we did our adult stress study, we compared the effects directly to getting either nicotine or amphetamine repeatedly. So we've only used stimulants for this experiment to ask this very question.
Mm-hmm.
The magnitude of the gene expression changes are similar. It's different pathways that are changed. So the answer is I think we're on the right track here. And we do know that being exposed to stress early sensitizes the brain to make it more sensitive to drugs, stimulants anyway. Mm-hmm. We don't know about other drugs. We do know that every psychoactive drug we've looked at, every class of psychoactive drugs we've looked at, so stimulants, depressants, anxiolytics, antipsychotics, antidepressants, all leave a footprint in the brain that looks to be permanent if you're a rat. And I can use an anecdote and say that my father-in-law quit smoking 40 years ago, but he says even now he'll wake up in the morning and say, I'd like a cigarette. So that's a long-term effect. Is he still an addict? Well, he would be if he started taking it. I'm sure he'd start smoking it, and he thinks so too. Mm-hmm. And a lot of people who are smokers will say it happened. I took a cigarette and I'm back, and I've got to stop all over again, and It's not easy.
But now the changes to the brain, like say if you would look at also get the spine count on neurons, and for instance the prefrontal cortex, which is one area that you looked at with great attention, would those changes be comparable in the epigenetic case as those of stress, or would the knock-on effect be different? Like if we take a mother rat exposed to different kinds of drugs, what is the impact on her offspring? Is it comparable to the stress case, or are the changes very different?
The changes are probably different. So I can do it from the anatomy and then make an inference from that. If we look at the effects of stimulants on medial prefrontal cortex, we get an increase in spine density. If we look at orbital frontal cortex, we get a decrease in spine density. What happens if we look at stress? We get the reverse. So what we see from stress is we see a decrease in spine density in medial frontal cortex and an increase in orbital frontal cortex. You might say, well, how do you account for that? Well, let's use a different class of drugs. Let's use opiates. If we look at opiates, it looks like stress. So it's going to depend on the drug type that you're using. So opiates look like stress. Stimulants look like the reverse of stress. In both cases, it's being changed, but clearly the mechanisms are not the same. We haven't done epigenetics with the opiates, so I don't know if it's more similar or not.
Right. But now, what you were mentioning is that if you look at these stressors as non-stressed or drugs, non-drug animals, that actually the morphology of the dendrite and the spines on the dendrite is rather different, in the sense that in, let's say, the healthy control case, you have a certain spacing of these spines that might be absent under either the stress or the drug condition.
Right.
Right. So how relevant is that for understanding then of these developmental and epigenetic processes?
Well, I think that one of the things it tells us is that subsequent experiences are acting on a different brain. So let's suppose your mom was a smoker, and forget about the effects of carbon monoxide. Let's just pretend the effect is nicotine. Your brain has changed. We've shown that. And you respond to experiences in a different way later in life. Same is true of stress. So your brain has changed. You respond to experiences such as drugs differently later in life. So we see these, what I'm going to call metaplastic effects, that sort of compound. And if you think about it, I mean, humans' life is not, you get a drug and then we kill you. It's one experience after another experience after another experience. And so this collage, if you like, of experiences are all meshing together to give you the final outcome. So this makes it in humans pretty darn hard to control. So you're going to expect huge individual differences. And of course, we see that.
But do you then look at development? Because in some sense, there's, if you want, a normal brain with sort of a developmental structure to the morphology of the cell, which is then a scaffold in which these future experiences are placed.
Right.
So what would that scaffold look like for the healthy brain? And just a cell morphology of a prefrontal or medial prefrontal cortex. You mentioned one thing, which is, for instance, the spacing between the spines, which I found interesting, because you were suggesting with that you would have, let's say, an optimal initialization of the dendrite with sort of an optimal spacing, so that future experience could be more easily linked into those structures.
So we've done experiments in which we've given animals tactile stimulation, or we've placed them in complex environments early and compared that to the effects of tactile stimulation or complex environments as adults. The effect is the opposite. So in the young brain, we have larger spacing between those spines, although the dendrites are the same length. So there's fewer connections, and it looks as though you can add connections much faster in this case. In adults, you see an increase in spine density in both situations. And it's clearly somehow changing the brain in a different way. What we haven't done is to try and get behavioral tests that are sensitive enough to see, is there a difference? And one of the problems is that the behavioral tests we use are designed to identify animals with brain injuries. They're not designed to do the experiments we're doing, so they're a bit naive in terms of what we're measuring. And that's an unfortunate problem at this point. And the same with the epigenetics. We haven't actually compared the two in the way we have with the dendritic organization, because the dendritic organization is cheaper to do and simpler to do than the epigenetics.
Right. But now you also mentioned on one of the spines, the generation of spines, and it's the pruning of spines.
Yeah.
And also this pruning process might be affected, because this is also a regulated process.
Yes.
It's not a random process.
No.
Right. So if we don't talk about some sort of epigenetic chain where gene transcription has to translate into, let's say, changes to the circuit, do you see one of the principles or one of the mechanisms we can use is the pruning process, and the other one is the generation of spines as two separate processes?
They're two separate processes. And the other thing we have to consider is which part of, if you think of the cortical layers, which layers change? Do the cells in each layer change the same? And my initial assumption was, well, of course, because it's the column is a functional unit. Well, let's actually measure that. Well, it turns out it's not true. So layers 2 and 3 and 5, for example, can change in the same way or opposite ways. What does that mean? I have no idea. It's just a fact that they're not, you can't predict from one layer to another layer. So it starts to get really complicated in there. So you're adding and subtracting spines. Are you measuring the same cells?
Mm-hmm.
Or are they different cells that are showing these changes? Don't know.
Mm-hmm.
Because the techniques we have don't allow us to identify the particular type of pyramidal cell it is, what its characteristics are, and measure those changes in spines. It's technically possible to do it. Now, we just haven't done it. But using molecular tricks, you can do it.
Mm-hmm.
And people will be doing that.
Right.
Yeah.
But now what we see here is indeed a brain that is hyperplastic at different timescales.
Yes.
And very sensitively tuned to changes in experience. But now in some sense, the experimental paradigms we use to probe that brain are by necessity simple, because otherwise we cannot control them.
Yes, that's right.
So to what extent are we actually getting the full picture here?
So we're not. So there's another way you could do it in principle, and that is to use resting state fMRI and look at the connectome, if you like, and see what in humans, and see whether or not you can extract things doing that. I mean, that's obviously very expensive, but I think that's the route that people are going to go to say, okay, can we actually do this in a more sophisticated way once we have the computing power to do it, and see if we see differences? And my guess is we will.
Right. But now the other link to the substrate is then again this FGF2, right? Which is sort of it's a growth modulator of the brain. And what you showed, however, is FGF2. It doesn't have a non-specific effect. It seems rather specific in its targeting of the brain. Right. So can you say something about that mechanism of the action of FGF2?
Well, it's related to where the receptors are densest. Mm-hmm. So if you look in visual cortex, for example, they're very sparse compared to prefrontal cortex or hippocampus. So the next question is, why the difference? And are there differences at different ages? Yes. The peak in the rat, anyway, FGF receptor expression is day 10, postnatal day 10. That turns out to be the age at which we get all kinds of wonderful effects that we don't see, say, at day 5, which is only 5 days earlier, but it makes a huge difference. So the FGF is specific in terms of where it's found and in terms of age as to when it's most highly expressed.
Mm-hmm.
Now, why? I don't know. It's obviously important for some reason. We don't know what's controlling that.
But that means there's a critical period also then for FGF2 and its impact.
Yeah.
And under normal conditions, how do you interpret that impact? Is that impact there to assist in, let's say, control a last growth push of, let's say, specific brain areas, specific part of the cortex maybe? Or what's the role of that?
Yeah, so the question you want to ask is what's happening around day 7 to 12? Mm-hmm. So 10 plus or minus 2 or 3. What's happening there that's different? And what's happening there that's different is migration is complete for the most part, and you're starting to get great cell differentiation and the beginning of synaptogenesis. And so it's a little bit, I'll use a metaphor that might not be great, but if you think about pruning a rose bush in the spring or when it's a young rose, if you do it at the right time, you get proliferation. If you do it at the wrong time, you might kill the rose. And so, and I don't know why that is, but presumably it's related to something like this, so that at the right time, it's primed to change. Let me give you another example, and that is if you kill the generation of neurons in utero at the right time, the brain can make up for that. It can, using X-rays, for example, the brain can make up for that and just overproduce as though nothing happened. That doesn't happen at other times in life. It's just at certain times the brain can do it. Is it just a party trick and it's just an accident? Maybe. Mm-hmm. In the case of the X-rays, that probably is. In the case of the FGF, probably isn't.
Mm-hmm. Yeah, because associated with that, you also showed that at least certain areas of the cortex you can lesion early in development, and it looks like they will essentially regenerate.
That's correct.
But that seems to be very region-specific.
It's very region-specific. It's specific to regions that have a lot of endogenous FGF2, which is interesting. Mm-hmm. Yeah, so if you do it in other regions that are really just millimeters away, it doesn't happen. But if you introduce the FGF2 subcutaneously, then at the right age, then it works.
So which regions are those?
Midline.
Mm-hmm.
So olfactory bulb, medial prefrontal cortex, and cingulate cortex.
Mm-hmm.
Why are those ones the ones that show the effect? I think it's because the nursery of cells in the brain is the subventricular zone is right under them. And so the cells can be produced there and migrate quite easily. They don't seem to migrate as well. So, for example, into more lateral cortical areas, there's a lot of stuff in the way. The signals that tell them to come may not get to them and so on. We don't know why that is. All we know for sure is that it's true. And if we introduce FGF2, we can get cells to go places they wouldn't have normally gone. How do you compare those cells to the endogenous ones? Well, at least in regions like the motor cortex, which are close by, it's pretty similar. If you're more lateral, we haven't really quantified it. I'm guessing it's going to be less similar. It's all related to mechanics of the progenitor cells getting there.
But the FGF2, you earlier also told us that it would enhance, if you want, brain growth, right?
Yes.
In a non-specific way, and also induced by the tactile stimulation.
Yes.
But now in this recovery study, let's say, we recover under the drive of FGF2, it seems very specific. So that seems contradictory in some sense.
It does.
Because on the one hand, it's like this a factor that leads to non-specific, let's say, complexification and growth of the brain. But under conditions of a lesion, it becomes very specific.
But so one explanation for that is that the brain is different. So the brain is producing all sorts of things in response to the injury, which makes it a different brain. And so when you add a spice to a dish that you're cooking, it depends on what you're starting with in terms of the effect of that spice, right? So if you see the FGF as a spice in a sense, a normal brain is different. The makeup of that brain is different than in the injured brain, which is producing all kinds of stuff that's trying to heal itself or make it worse, whatever. But there's both going on.
But that would mean that there's another system yet again that is regulating that uptake of the FGF2.
Yeah, and it may be it's glial cells. So in response to the injury, you're going to see the production of both astrocytes as well as microglia. And we know that the astrocytes, at least, are producing all kinds of chemicals. And they produce FGF2, it turns out. So what do the microglia do? Are they producing things that are having an effect too? We don't know the answer to that. So the brains really are different. So we shouldn't be so surprised that the effect of any compound is not going to be the same. The lesion brain is different.
But would that then mean as with the tactile stimulation, I generate FGF2, do I in parallel then drive a mechanism that controls its uptake? Or do you think it's more unspecific than that? Because like maybe what you have to with tactile stimulation, I must drive up my FGF2 and I must start to engage my glia cells to make sure they sort of create the conditions in which uptake can take place effectively.
Yeah, that could very well be.
But how do you think about it? And do you have the simple interpretation, or do you think it's really more complex than that?
Well, in the injured brain, it's clearly more complex. In the experienced expectant case, it may be that simple, that the brain is expecting experiences, and when it gets those experiences, these things happen. The FGF2 goes up. Think about the visual system. The visual system is expecting visual input. Doesn't require very much to tune your ocular dominance columns, the stuff that Colin Blakemore did all those years ago. I don't recall the total number of minutes, but minutes of visual experience is sufficient to satisfy the brain and prevent the effect of ocular closure.
Right. So now, so this is largely done on the rats, and there are some links to human behavior, largely. Do you think that these lessons that you've now extracted from the rat brain and rat behavior about epigenetics generalized directly to the human case? Is the human case maybe even more susceptible to these epigenetic factors or less?
I would say the human brain is more plastic. There are more neurons. They're more densely packed than any other species, and certainly much more densely packed than rodents. So yes, I think that it's going to generalize, but I think the effects will actually be larger for that reason.
Mm-hmm.
It also has more white matter. So the gray matter, white matter ratio is totally different in humans than it is in rodents in particular. And so what does that mean? Well, when we're studying treatments for stroke, I'm sure that it makes a big difference because you can have huge effects of white matter injury in humans, not so much in rats. So we have to keep our eye on these differences that are fairly gross.
Mm-hmm.
And of course, humans have regions that rats don't have. I mean, no rat talks, at least none that I've met. And so they don't have Broca's or Wernicke's area. So what effect are these experiences having on these language-related areas? We're the only animals that produce music in the sense that we mean it, not birdsong.
Mm-hmm.
So what does that mean? We know that music has a huge impact on the brain. So, for example, if we looked at cognition in older people, so say over 65, there's going to be a decline. And those people who are also musicians or who not professional, but who learn to play instruments early, there's benefit. So memory's better, attention's better, and so on. And the older you get, the bigger that effect is.
Mm-hmm.
So there's a reduction in, say, dementia in people who have music. What's it doing?
Mm-hmm.
It's not changing, as far as we know, non-human brains, but it's having a big impact on ours.
Mm-hmm.
So
right.
Yeah.
But also you mentioned that in some sense, there might also be, let's say, unwanted side effects of some of the medications we use actually in a standard fashion. If you mentioned the Prozac example, that initially might look like a great idea to drive also brain development or brain adaptation, but maybe from an epigenetic perspective, it might be the wrong thing to do.
Yeah. So the story there is that we expected that fluoxetine, Prozac, would actually stimulate brain growth and we get bigger brains. And we didn't. We got smaller brains and brains that are less plastic. That we didn't expect that. And the doses are the same doses in the amount in the blood is the same that women would be getting, or the babies would be getting. So, yeah, that turned out to be, you can call it a side effect, an unfortunate consequence of the treatment of anxiety or depression.
So right now it is prescribed to pregnant women as well.
It is. Mm-hmm. And there is a suggestion that it should not be prescribed for things like anxiety. Try some other drugs. Mm-hmm. But one of the drugs that's used more in Europe than in North America is valproic acid, or valproate. It's used for epilepsy as well as anxiety and so on. And that turns out to have a link to autism. So the best model for developing autism in rats is to use valproate. That's a very unfortunate side effect. Mm, right. And in England in particular, have been studies done showing a huge increase in all kinds of neurodevelopmental problems in women who were given prescriptions for valproic acid for one of many reasons.
Mm. So there's an important lesson there.
Absolutely. Yeah.
But now, so you worked with rats for decades, mm-hmm. and
I've worked with people too.
And you talk to people as well. But,
yeah.
But in some sense, how well do we understand rat behavior and the rat experience of the world?
So Ian Wishaw and I have a book called The Behavior of the Laboratory Rat. It's edited. And so I think we understand a lot about the behavior of rats compared to the behavior of mice, mm-hmm. or cats for that matter, which aren't used much for behavioral studies anymore. So we know a lot about the behavior of rats. The problem is the sophistication in measuring the behavior isn't always there. So there's two ways to do it. One is to use what I'm going to call an endpoint measure. So if we're looking at something like skilled reaching, how many pellets did you actually successfully reach for? But we can also ask the question differently and say, are the kinematics of the movements the same or are they different? So we can see in animals that have adult strokes, we can give them treatments and their endpoint measure appears to be normal, but the way they're doing it's quite different.
Mm-hmm.
And so I think, yes, we know a lot about the rat behavior, but the sophistication of the average researcher in terms of how you measure it still needs to be improved.
Mm-hmm.
And
right.
I'm guilty too. So I mean, it's.
But then where do you see the future? Do you think that these attempts to, for instance, start to use virtual reality with these kind of animals is a step forward, or you think that's not helping? By creating, in that sense, also more dynamic and more complex environments?
So do you think you can use virtual reality in rodents?
Well, it's already happening, right? People do it. People have rats run on little balls that float in the air, styrofoam balls.
Yeah.
And with that, you then drive a virtual reality display of an environment. So they run through the environment. People are measuring place cell responses or grid cell responses using these setups.
We know that it works in people. So if you have stroke patients and who have motor problems, by using virtual realities, you can get quite dramatic improvements. We don't know why they're improving, but we know that that's happening for sure. People haven't really done those kinds of experiments in rats looking at the effects of, say, stroke. And can you use virtual environments to enhance it? Don't know.
Yeah, we've been treating over 500 patients with that approach here.
Yeah.
With really good outcomes.
Yeah.
So on humans, it works great.
Yeah.
You know? But now, so you've been in this field now for a long time. You also started life as a so-called rat runner, or you came from another direction into that? My
master's degree was in ethology, so it was in animal behavior, and I was looking at rodents. When I was doing my PhD, I worked with a variety of animals, including cats and hamsters, gerbils, and rats. And then pretty much, I then spent time at Montreal Neurological Institute studying people with brain injuries and trying to take the lessons I'd learned in terms of studying behavior in lab animals to people. Could you actually score their behavior and see similar effects? And the answer was, yeah, you can. I didn't have the opportunity that I had at the Montreal Neurological Institute because there were so many patients when I left and went back to Alberta. So it was pretty much back to rats again. But I think my experience at the MNI in terms of generating new behavioral tests that were based on my studies, mostly of cats, more so than rats, was successful. So it encourages me that, yes, there's going to be a good transfer.
Right. So now, given all this, the experience you have in the study of rat or animal behavior or human behavior and the brain, if you would like to follow in that tradition, what's Brian's law that we should adhere to?
One of them, I think, is what does this mean for people? So I think that it's really important that people who are studying rats know something about the human brain. They know something about the effects of experiences on the human behavior, because if you're doing it in a vacuum and you're just studying rats, it's useless. And you really need to have this broader perspective. And this was one of the reasons that Wishaw and I wrote our textbook, Fundamentals of Human Neuropsychology, which had more animals in it in the early days than in the 7th edition, but it really is trying to take the principles and understand how the human brain is working. I think you have to keep that in the back of your mind all the time, because if you don't, you end up studying epiphenomena, and you're losing touch with what's important.
Right. So then 5 years from now, we're going to go visit your lab, and we're gonna check whether a prediction that you're going to share with me now is actually confirmed or not. So what's the key prediction that you would like to commit yourself to today? That is the most important one to make progress in your field in this time frame of 5 years.
Well, at least not in my field, but in my lab, would be understanding metaplasticity. That is the interaction of experiences, how they combine together to give you this phenotype down the road. Which ones are more important? Does the order make a difference? And so on. So that would be sort of the final push. That's the one. The other one is adolescence, because our emphasis has been in adults and in developing animals. But where are some of the biggest changes, especially in prefrontal cortex? Adolescence. We've begun to look at the effects of treatments in adolescence, and they're different. And so let's just take this isn't our work, this is more general. If you look at the effects of marijuana consumption, if I can use that term, or nicotine consumption on adolescents, the incidence of psychotic episodes in the 20s is way higher than it would be in you if you started taking either of those drugs now, post 30 or so. So why is that? There's got to be some sort of change, epigenetic and otherwise, in response to the adolescent experience. Well, the adolescent brain is changing so rapidly. And I gave you the example this morning of in the peri-adolescent period, as the prefrontal cortex starts to prune, you're losing 100,000 synapses a second. That's a huge change in the brain. So any experiences that you're going to encounter while this is going on are going to have a huge impact. It's like a second sensitive period. You've got the early one, say 0 to 3, then this other one, say age 10 to 16 years. That's really going to determine who you're going to be. Mm-hmm. So we don't know anything about this. So that's the two things. So one is metaplasticity and the other one is the adolescent brain. We need to understand it.
Very good.
Yeah.
Well, Brian Kolb, thank you very much for this conversation.
You're welcome, Paul. Thanks for having me.
Sure. Well, wasn't that fun?
That was it was fun because you're forcing me to think about things in a different way because you're coming at it from a more human perspective, if you like. Yeah. And we tend to, well, I'm guessing you do too, in your own research, you have your favorite hypothesis and you're doing this, and then occasionally something happens over there and you say it's trivial.
Exactly. Yeah, right.
And you just keep chugging along. Mm-hmm. And so that's why I'm giving a lot of public talks because of Fraser Mustard I mentioned this morning and the importance of educating the public on early experiences and whatnot. But some of the questions I get from the public are sort of, whoa. Mm-hmm. So in Canada, we have a very bad history of dealing with our Indigenous people. And one of them in Canada, the Americans had a different solution, and that was to kill them all. Mm-hmm. In Canada, we said, no, let's just take away their past and put them in what we called residential schools, and we'll make them white. Mm-hmm. It didn't work.
Right.
It made them worse. It made it worse. So you end up with these horrible schools run by the Catholic Church, in which these kids were maltreated, and now we don't have them anymore. The last ones were closed in the early 60s, but the effects of those schools is still there.
Still there, yeah, right.
And I gave a talk in northern Alberta, and there were a lot of natives there, and gave the sort of simple spiel. And this one elder said, let me get this right. Are you giving me an explanation for why residential schools had such a profound impact on us? And I said, yes. Why hasn't anybody told us this before? I said, I'm here, I'm telling you. Yeah. You're not to blame. Mm-hmm. We now understand one mechanism as to how these residential schools could lead to all sorts of drug abuse. So let's correct it. Mm-hmm. Let's correct it. Let's get by it.
Right.
And figure out ways in which we can reverse the effects.
Yeah, but
because these are cross-generational effects.
Of course.
Yeah.
And that might be still a long shot, right, to now have an influence on that. It'll take generations.
Of course, yeah.
No, but it's also from my perspective, what we do here, that we definitely have a strong focus towards the society in everything we do, especially to stay on track, to stay focused, to stay relevant, because otherwise it's very easy to just get sucked into your rabbit hole.
Oh, for sure.
And, you get lost forever.
And like Alice in Wonderland, you're just sort of wandering around.
Exactly. It's all beautiful, right?
That's all beautiful.
Yeah, exactly.
Yeah.
And so we're in the clinic very prominently, a lot of stroke work we do, also farming out to other pathologies. Now, Parkinson's disease we're looking at, and palsy. I think cerebral palsy is an important target. Look at education, because I think the educational system is completely broken. We have to...
Is it specific to Spain or?
No, Europe. It's all European projects that we're doing. But in all cases, think more...
The CSN podcast was produced by the Convergent Science Network of Biomimetics and Biohybrid Systems.
Do whatever you want.
Project funded by the European 7th Research Framework Program.
Huh? Yeah, I will. Don't worry. So I was wondering about this distinction between learning and epigenetics, right? And this has been quite a debate. And in some sense, we can impose also the question, okay, are we back in a more Lamarckian view on evolution? Because now we were looking at experience-dependent impact across generations, right? So what's your position on that?
Well, I think in part you've hit it on the head. We are getting a little Lamarckian, and Lamarck didn't have any way of measuring. What he thought he was getting was something different than it turns out it is. But he had some truth to it. But we do know that behavior itself produces profound effects on the brain. So this is most non-psychologists would be surprised at this, I think, that behavior changes the brain. They're going to think the brain changes behavior. Mm-hmm. Well, that's true as well. So you've got this interaction. So the experiment that I was mentioning is that if we train animals on a whole bunch of behavioral tests, cognitive, motor, and so on, and just look in the brain, do we see change? We see changes all over the place. Okay, what if we give them something like nicotine or amphetamine? We see changes all over the place, but they're not as big. If we give them those drugs and then later put them on do all the behavioral tests, we see a much bigger effect. So this is the metaplastic effect. Mm-hmm. So I think we've underestimated how big the effect of actually learning, if you like, is on the brain. It's really producing profound effects. And this takes us back to the children and their vocabulary early on. They're learning all of this stuff, and it's producing these profound, long-lasting changes. So when we sent this paper in, showing and tried to make the point that the behavioral training was as big or bigger in its effect than the drugs, the reviewer said, well, this is trivial. I mean, behavior isn't all that important in understanding changes in the brain, and he didn't get it at all. So we obviously didn't describe it effectively. But for psychologists, it's obvious. You're going to say, well, of course, it's going to change the brain.
But now the point then is also that we would have a situation where, let's say the mother has a certain exposure. This carries over to the offspring. But you could also argue, well, it's not necessarily Lamarckian, because in the genome, there are, let's say, different phenotypic programs. And they are just triggered in an experience-dependent way. Right? Is this how you think about it?
Yes. That's how I think about it.
Right. So then, so if we then can be configured along these different epigenetic programs, how big would that repertoire be? Is it only like stress, no stress, or do you think this is a much more high-dimensional space?
Oh, it's more than one-dimensional for sure.
Okay.
Yeah, because the stress itself is producing changes in behavior. It's producing changes in cognition, and so on. I'll call that a behavior as well, but people tend not to think of it that way.
Right.
And so, yeah, we're getting a multidimensional change. From all, yeah.
Okay, fantastic. Thank you. That's the one I still wanted to have.
Okay.
All right, we're done. Thank you.
Okay.
Noted.
The CSN podcast was produced by the Convergent Science Network of Biomimetics and Biohybrid Systems, a project funded by the European 7th Research Framework Programme. For more interviews, recorded lectures, or upcoming conferences in the field of biomimetics and biohybrid systems, go to csnnetwork.eu. And thank you for listening.